do today is to talk about some aspects of COVID-19 vaccinations and kidney transplantation. Now, COVID-19 is such a big topic, and there has been so much literature that came out on this that PubMed, etc., have gated COVID-19 results away because there's just so much of information out there. So I'll try to distill it down, but I will try to restrict myself to active and passive prevention of COVID-19 by way of raising antibodies. So before we start, my disclosures: I have speaking engagements or consulting arrangements with CareDx, AstraZeneca, and GSK. None of them are relevant to the talk today, more or less. So there's been, I don't think in recent times, an endeavor like our response to COVID-19. This is one of the worst pandemics that we are witnessing. It's probably not a once-in-a-generation phenomenon. It is a once-in-a-century phenomenon. We knew nothing about this in December of 2019. The only thing we could figure out was it's a contagious disease, probably respiratory, and so all we had at that time is masks, the 6 feet social distancing, and the hope that we might get a vaccine. At that time, we did not know the structure of the virus. So this is December 2019 when the first case was described in China. We had not had a case in Europe at that time. We had not had a case in the United States. And this is the virus, this is the villain, right? So this is an enveloped RNA virus, and it is a very, very simple creature. It's one of the larger RNA viruses, and the important thing to notice are the 4 proteins that mark its antigenic structure. And the 4 proteins are the spike protein, the yellow little spiky things that you see come out of it. There we go. I think we can probably. So I'm going to use this screen for my pointer. Apologies to this side because that's a little farther away. So these spiky proteins are the spike proteins. You're going to hear a lot about them, and we'll talk about them a little bit. Then you have the membrane protein, which is associated with the membrane of the virus. You have the envelope protein, which is associated with the envelope of the virus. And you've got the nucleocapsid protein, which is supporting the RNA motif here. So that's all there is to it. You know, when we look at the spike protein, this is a X-ray crystallographic structure of the spike protein. This is the business end which binds the ACE receptor. And when we started talking about COVID and we realized that it gains entry into the cell by engaging the ACE2 receptor, many of fruitful discussions happened about whether we should continue ACE inhibitors in these patients or not. But at that time, everything was a question mark. So I'm going to just spend a little bit of time on the spike protein because you're going to hear a lot about it. So imagine this spike protein cartoon over here. It looks like 3 golf clubs stuck in the bag. So this trimeric structure, so the 3 little golf clubs are all made of 2 regions. The S1 region is membrane distant, away from the membrane, and has the N-terminal domain as well as the RBD. Now you're going to hear about the RBD a lot, so I wanted you to see what that was. RBD is the receptor binding domain. It is this area that actually goes in and attaches to the ACE receptor, H2 receptor on the cell surface membrane. And once that happens, there's a conformational change. So the S1 segment breaks off, and there's a conformational change in the S2 segment, which starts looking a little different. And then that attaches the virus to the cell. The virus RNA goes inside the cell, takes over the cell machinery to make more RNA, more virions, destroys that cell, and goes to the next one. And that's how the virus causes pathology. Now, if you could block this, right, you could stop this entire sequence of events. And therefore, most of the antibodies that we have tried to generate, actively or passively, actually target this RBD, receptor binding domain, in an effort that we won't have this ACE2 and spike protein interaction, and the virus won't get inside the cell, avoiding a lot of heartbreak. So that's kind of the basic theory. So now we are looking at December of 2019. Believe it or not, within a few weeks of the first described case of this virus, the entire genomic structure was published for everyone to see. And so the next day, the NIH started working on a vaccine. So I'm going to talk about how a vaccine really works, because there's been so much of conversation around this. So you got these antigens. Antigens are molecules that would provoke an immune response. So you stick them into the skin and inject them into the muscle so that they're slowly released. And antigen-presenting cells, whose job it is to scavenge against any infection, pick up these molecules and then they present them to the immune system. Now, we're going to make this a little bit more complex as we go through, but what the antigen-presenting cells are doing is they're saying, this guy doesn't look like my own people, so I'm going to see if there's a problem. And so they pick up these molecules through these PRR, pattern recognition receptor molecules, and start chopping them up inside and presenting pieces of these proteins to the immune system. Now, if the immune system says, nah, this looks like my own, then there's no problem. But if the immune system says, hey, this looks like a foreign agent, then different types of cells start getting into action. There's two different types of T cell tribes, the CD8 T cells, which are like the infantry soldiers. As soon as they get the word, they go and start attacking anything that's displaying these molecules. And then the more well thought out, detailed response comes from the CD4 arm. They are going to then not only generate more CD8 cells, but also start talking to the B cells, and the B cells make antibodies. So we'll kind of go over this in some detail. Now, there are other molecules inside this vaccine called adjuvants. And adjuvants are molecules that enhance the immune response. So when you've got the antigen of interest and the adjuvant in there, then the adjuvants are going to ensure release of certain cytokines, interferons, etc., which then make the response a little bit more robust. Now, I said I'm going to make it a little bit more complicated, so this is going to be probably the only more complicated slide from an immunology standpoint. Not really, but one of them. And so essentially, we are going to look at a lymph node here, right? So this is a lymph node. This is the outer area of a lymph node, and this is where the antigen-presenting cells are sitting around. And this is where naive B cells are sitting around. They're waiting to be called up for service. So when the antigen-presenting cells see this vaccine material, so if you've injected it in the deltoid here, in the draining lymph nodes in the axilla, this is happening. And what happens is the antigen-presenting cells pick up this antigenic material, talk to these naive B cells. Once this happens, the naive B cells say, hey, this is something I need to work on. They kind of start migrating towards the center of the lymph node. And there's a critical step that happens. I told you that the T cells talk to the B cells. So once the CD4 T cells are activated by the antigen-presenting cells themselves, there's a particular class of them called TFH, follicular help cells. These are very, very essential in leading these B cells to what is known as a germinal center reaction. And so what these do is they provide what is known as co-stimulatory support to the primed B cells, and that leads the B cells to start multiplying. So when you have an infection, when you have mumps, when you have measles, I don't know how many of you have seen a case of mumps or measles, there's associated lymphadenopathy. The lymph nodes get swollen. When you have a sore throat, sometimes your lymph nodes in the neck get swollen. That's what's happening over there. There's a germinal center reaction. All of these B cells can then generate three different fates. They can cause short-lived plasma cells, which make antibody. They have primordia of memory B cells, which can launch a faster recall response, or they can go back and enter the germinal center reaction again. And this is very important because this is where the B cells undergo a process called affinity maturation and somatic hypermutation. The repertoire on the surface receptor of the B cells is getting refined. It broadens up a little bit. So the more B cells go through this, the more they're able to attach to antigens which look like the antigen of interest. And eventually what we get is memory B cells, and I'll explain to you why this is important, long-lived plasma cells, and some B cells which go back and keep getting recruited into this response. So if you've received childhood vaccination like mumps or measles or rubella, you can actually note antibody present in the blood decades later, right? How does that happen? The reason it happens is you've got these long-lived plasma cells sitting in the bone marrow, and they're chugging out this antibody, and they can live for 10 years, 20 years, 30 years. That's why when you're looking at tetanus, they say get a booster every 10, 15 years, because that's how long these antibodies are going to last. That's also how long some of these memory B cells are running around, and they can, as soon as they see that antigen, as soon as they see tetanus, as soon as they see measles or rubella, they can turn into plasmablasts, plasma cells, and lay out sheets of antibody. So this is the goal of vaccination here as well. This is a response that could be seen against an infection, but when you introduce vaccine material, the same thing happens, the same process happens, and therefore if somebody's gotten vaccinated, now they've got COVID antibody in their blood. Where's that coming from? Because the vaccination was months ago. It's coming from the fact that you've got long-lived plasma cells sitting in the bone marrow, and you've got memory B cells. So if, God forbid, you're exposed to COVID now, then your immune response will turn on in 48 hours. A bunch of antibody would start coming out, and hopefully you won't get as sick. And that's why we are not seeing people get as sick as before, because we are all, or most of us are now, vaccinated. So how do you get this material to the immune system? That's the issue, right? That you've got to get some of those antigenic signatures to the immune system so it can make antibodies against the virus. So conventionally, we have actually done live attenuated viruses or inactivated viruses, but in case of COVID, that was a little bit of a problem. We didn't know. It sounded like a highly contagious virus, so dealing with it in the laboratory was a little bit of a problem. So with live attenuated viruses, you sometimes run the risk that the disease itself may come out, and therefore you can't give live attenuated viruses to any of our transplant patients. Everybody knows that, right? So we don't want to do that. And then inactivated viruses, since you have heat inactivated or chemically inactivated the viruses, sometimes the antigenic signature changes, and the thought was that we might not be able to provide adequate antigenic delivery. So now comes in the miracle of modern medicine, and it's kind of, there were some technologies that were at the cusp of getting getting approved. So we brought them to bear. And so the way this was done was to utilize nanotechnology and different vehicles for this, using lipid bilayers, so lipid nanoparticles, or using lipid conjugates to proteins or peptides, or creating virus-like particles, VLPs, etc., etc. The idea is that these little particles are going to pick up the antigenic material and take it. So you inject it subcutaneously, and these would then ensure delivery of it to the antigen-presenting cells, so that the antigenic material is not destroyed in the process. It is delivered at a sustained rate. And importantly, these lipid conjugates, etc., which are acting as the nanoparticles or delivery vehicles, actually have adjuvant qualities to them as well. So they help in generating the immune response. So there's three different genres of antigenic material delivery. All these 7 ways have been tried in COVID. Some of them are not being utilized. We're not using live attenuated virus, but we are trying to attempt primarily delivery of virus to the antigen-presenting cells, delivery of protein material to the antigen-presenting cells, so that the antigen-presenting cells can do it in vitro. We are trying to directly get protein to the antigen-presenting cells by using protein subunits, or VLPs, or inactivated virus. There is one inactivated virus. I think the Sinopharm vaccine is the inactivated virus, which is not being used in the United States. So these are the 3 different ways: in vitro protein delivery, direct protein delivery, and live attenuated viruses that have been thought of as potential vehicles for delivery of antigenic material, either through the RNA vaccine, DNA vaccine, virus-like particles, etc. The idea is for all of this antigenic material to get to the antigen-presenting cells so it can engage the immune system. That's all there is, but this is an engineering miracle that has happened. So what happens when you stick the antigen into the skin, into the muscle? So the antigen comes in and it goes to the antigen presenting cells. So the first line that you're going to see is it's going to talk to CD4 and CD8 cells. CD8 cells are the assassins, the killer cells, not natural killer cells, but they kind of kill on contact, and so that's your first line of cell-mediated immunity. The CD4 cells generate different types of populations, but they are also going to stimulate CD8 development, but they are actually going to invoke the antibody arm, and so some of the CD4 cells convert into the T follicular help cells that we talked about a little while ago, which talk to B cells and start the B cell response to make plasma cells, which make antibodies. All right. Now, I'm going to talk a little bit about the germinal center response in this, that if the germinal center response doesn't develop, so if there's not enough help between B cells and T cells, if this does not happen well, then what we can see is that instead of proliferation of B cells, you start seeing those B cells dying. And the reason I wanted to put this slide, which kind of looks out of place here, is that we'll come back to this when we talk about our transplant patients. And eventually, the second line of events that happens here is that now you're going to develop memory. So the memory, immunologic memory, is both on the T sides and on the B sides. What does that mean? So I told you a whole sequence of events that you get antigenic exposure, then the antigen presenting cells picks it up, then it talks to T cells, it talks to B cells, the T cells start developing, the B cells start developing, and eventually an immune response develops. This takes time, usually about 5 to 7 days. But when you've got memory in there, when you've got memory cells in there, T cells and B cells, within 24 to 48 hours, you start seeing an immune response. And so based on these technologies. These are the vaccines that are the finalists. You've got the Pfizer vaccine, which is an RNA vaccine. You've got the Moderna vaccine, which is an RNA vaccine. These are all approved under the Emergency Utilization Act by the FDA. You've got the AstraZeneca vaccine, which is an adenovirus vaccine on chimp adenovirus cells. This is not approved in the United States. You've got the Johnson vaccine, which is a human adenovirus vaccine. This is approved in the United States. And then finally, now this is the only vaccine I'm not going to talk in detail about, the Novoxoid. This is a protein-like particle, so there's protein material in there, and it's a slightly different concept. It's one of the different antigenic material delivery genres. So when you've got somebody who's uninfected and they get the first dose of the vaccine, they develop plasma cells and memory B cells. We're just talking about the B cell side over here. So there's a response, and there's neutralizing activity, and then there's neutralizing breath. Let me kind of explain to you why these three different things are important. The response is you've got antibody, right? Now, does production of antibody mean that this is actually going to kill the virus? That's your neutralizing activity. That's what we care about. I don't care if you've got tons of antibody, but it is not actually going to do anything to the virus. And neutralizing breath becomes important when you're looking at variants that are developing, because we know from our study of the flu that there are antigenic changes. So the virus may mutate into something quite different, and your antibody may not actually be effective. So remember, we were talking about how the antibody is supposed to attach to the RBD, the receptor binding domain of the virus, right? Now, if the receptor binding domain changes its cloak, then the antibody is not going to attach to it. And we saw this with the monoclonal antibodies that we were using to treat the virus with, and the Omicron variant, where they were no longer effective. So, we were using bamlanivimab quite frequently. We're not using it at all now because of the Omicron variant. Now, when the same individual gets the second dose, now you've already got some priming activity. You've got memory B cells in there. There's a bigger response. The response magnitude goes up. The neutralizing activity goes up. And the neutralizing breadth starts increasing. And the reason I'm showing you this is because that's where the booster concept comes from. And that's where the answer is to deal with all these variants that we're dealing with. Now, if somebody's previously been infected, they already have plasma cells and memory cells because they've actually been vaccinated through the infection route, not through the vaccine route. But even in those patients, when you give them an additional dose, additional booster, their response magnitude, neutralizing activity, and neutralizing breadth goes up significantly. And when you give them an extra dose, this gets better. So essentially, sometimes people ask, well, I already got the COVID. Should I get a vaccine or not? The answer is yes, because you're going to derive a benefit from that. And this is why this is important, the last couple of slides that I was showing you, that as soon as the virus came out, remember the first case of the virus was December of 2019, so that's a short 2.5 years out. And as soon as the virus came out, we started getting reports of the variants. Different parts of the globe started reporting variants. Now, what is a variant in this situation? A variant is where the RBD, mostly, has started changing its antigenic signature. So the concern and the question is our vaccine going to still be effective when there's a variant in play? So this is a complicated slide, so I'm going to try to walk you through it. So essentially, on the top panel, we see wild type. So this is the virus that has not mutated, the Delta mutant, and then the Omicron variant. So remember the Delta variant, we've all forgotten about it, right? It came out about a year ago. Now everybody talks about Omicron. And all that's happening is that golf club, the inverted golf club we were talking about, there are mutations in that area, in the RBD of the S1 part of that golf club. And so the wild type which is closer to the Wuhan strain, right? The Delta is one set of variations, Omicron is another set of variations. And so what they're looking at here is you're giving them the vaccine series and then a third shot. And what you're trying to figure out is what happens to their ability to launch antibody. So in all of these, when you give them the third shot, the antibody levels start going up. Even in the Omicron, the antibody levels go up. But the question is this antibody actually working or not? This panel, what you're seeing is a correlation between the ability to neutralize the virus in the wild type versus Delta. The black circles are the guys who have been vaccinated and then boosted. The white circles are the guys who have just been vaccinated, right? So these guys have received more doses, and the booster could be homologous, that is, if they got the Pfizer, they get the Pfizer booster, or heterologous, where if they got the Pfizer vaccine to begin with, they get the Moderna booster. And so what we see over here is a correlation, some correlation in virus neutralization activity in the Delta variants in the unboosted population. But really, when you get to the boosted population, the Delta variant was neutralized as effectively as or close to as effectively as the wild type in these patients. Similarly, in the Omicron, the unboosted people actually did not neutralize effectively, but the boosted people actually did. See how it is clinging the correlation bar there. And also, again, in Omicron, what we see is that the virus neutralizing threshold is a little high. So you need more antibody, which is also shown in this receiver operating curve over here. So in these curves, what you want to see is the curve to be as close to the northwest window as possible. So the black line over here is wild type, the middle line is Delta, and the magenta line is Omicron. And so as your virus level goes up, the ability to neutralize goes up. So what this is actually telling us is that if you get extra boosters because of the affinity maturation and somatic hypermutation principles we were talking about inside that lymph node, the antibodies vary enough to kind of counter some of the mutations that are happening on the virus surface. So how do we fare? Most populations do okay as far as neutralization activity is concerned. So that's what we care about, right? I said that we want antibodies that are going to neutralize the virus. So we see this in elderly patients. We were concerned that they may not launch a response. They do. Pregnant females can launch a response. Autoimmune disease patients, some of them are on immunosuppressive medicines, they do okay. People with controlled HIV infection do okay. But the two tribes of patients who don't do well are cancer patients and patients with kidney transplant. And that was very, very worrisome because this is a very at-risk population because they're immunocompromised. So this is from the Strasbourg group from France. And what they did was they looked at COVID-naive patients and they saw, well, what happens if you give them two vaccines, right? And we knew nothing about this. The first vaccine became available, December, late 2020. So this study was done in 2021. And this is against the classic variant, the vaccine was. And so what they found was that very few patients, somewhere in the magnitude of about 30 to 40% of patients, actually started developing a response. And those who developed a response, their antibody titers were not very high. Very, very disappointing. And different types of immunosuppression, if you were on steroids, you did poorly. If you were on tacrolimus and an MPA formulation, you did exceptionally poorly. So that's kind of intuitive. You've got immune suppression on, and we were thinking, well, they're not able to launch a response. Kind of half expected it, but there is more to this story, and this was seen in our Johns Hopkins group as well. This study was published in JAMA, and what they looked at was, so the two panels A and B are showing results from two different types of EIAs. This was their in-house EIA. This is the Euroimmune EIA. All these different lines are the trends in individual patients between the two boosters, and what they found was about 40% of people are responding. So less than half of your transplant patients are going to generate any meaningful antibody response after 2 doses. Very worrisome, not good. This is from the Toronto group, same deal. 5% of people generate a response to the anti-RBD level. RBD, everybody knows what it is, the receptor binding domain. That's the outer part of the club. This is what we care about, right? And then what we care about is neutralization capability, again, very low. So our transplant patients who are getting 2 doses of the vaccine are not doing well. That got us very, very worried. This is the T cell response, so CD4 T cells, the ones that kind of orchestrate the response. Somewhat of a modest change in them, but the CD8 cells, which are the soldiers that go out and kill immediately, hardly a response. Why would this be, right? So this is a very, very interesting study, and I told you that when you inject the vaccine, it goes into the lymph nodes. So this group actually studied lymph node aspirates in patients with kidney transplant and 10 healthy donors who are not sick, just gotten the vaccine. So you get the jab, and then they start taking out lymph node aspirates from this side and the other side to see actually what's happening inside the lymph node. So V1 is the first dose. V1 is the visit for before the first dose. You get your first jab, V2 and V3. This color coding, I'm not going to walk you through the entire thing because I've done that for you. It took me about an hour just to understand the information, and I've just clipped parts of it. But essentially what they're telling us is that the germinal center reaction in these patients doesn't happen. So on the side of the jab, when they drew plasma cells or RBD specific memory B cells or RBC specific mononuclear cells, they didn't see any in those patients who had a kidney transplant. They did see them in patients who were healthy donors. So there's something terribly wrong here, and that is why we are not seeing the immune response. And so this is the concept that's very important to grasp in our kidney transplant patients. So this is what you expect. You expect a germinal center reaction, either in case of an infection, which sometimes disrupts it too, but especially when you're doing the vaccination, that is lost in kidney transplantation. And therefore, the amount of antibody that is being produced, the neutralization capability of that antibody, and the breadth of the response, the ability to counter variants, that's all very suspect in kidney transplant patients. So this is a very elegant study by by a group that calls themselves the Renal COVID vaccination group. So essentially what they did was they took some controls. They got patients with CKD4 and 5. They got patients on dialysis, and they picked up kidney transplant patients, right? So when we were looking at these responses in kidney transplants, we said, well, they're immunocompromised, but so are dialysis patients. They're immunocompromised as well. So essentially, what we see is when you give them the first dose, right, a huge number of controls respond. We know that. A huge number of CKD4s and 5s respond. A fairly large number of dialysis patients respond. When you give them the second dose, almost everybody who's a control responds. almost everybody who's on CKD4 and 5 responds. Everybody on dialysis almost responds. Very, very few who don't. But look at what happens in kidney transplant. Your initial response is suspect. About 50% eventually respond. That's the number that we got from the French group. That's the number that we got from the Hopkins group. And then there's a fairly healthy area of people who are not responders, not good. And the antibodies and the ability of the antibodies to neutralize. So this is looking at kidney transplant recipients plaque reduction neutralization assay, which is a measure to see if the antibody is actually killing virus or not. And essentially people who've had kidney transplants fare very poorly. And the ability of the antibody to reduce plaque is directly correlated to the amount of antibody. So that makes some sense as well. So essentially, that was very worrisome. Now, I want to spend a little bit of time. This is the same study, and what we are looking at here is healthy controls. This is their antibody response. But if they're on immunosuppression, so panel A is prednisone, calcineurin inhibitor, and MMF, the ability to generate this antibody goes down. If they're on lighter forms of immunosuppression, so this panel here, panel B, is prednisone plus calcineurin inhibitor, but instead of MMF, these patients are on azathioprine, you actually produce more antibody. And it turns out that the severity of immunosuppression affects this, but especially MMF is a bad actor. So is CD20 depletion. So if somebody has received molecules like rituximab, this is a study in multiple sclerosis patients who were on CD20 depletion through a molecule called ACD20. And essentially what they did was they gave them 2 jabs of the vaccine in the patients, 20 patients who were on CD20 depletion and 10 controls, and then collected their blood at 5 time points. Obviously, starting, they had no antibody or RBD antibody, but as time goes by, the controls launched a good response, but the patients who were on CD20 depletion actually had poor responses all the way through. And again, in CD20 depletion, their memory B cell generation inside the lymph node is affected. It turns out that if you've been on CD20 depletion, if you've received rituximab, it can be months before this response is generated. Mycophenolic acid we talked about is a bad actor, and there's a linear correlation between the dose of MMF. So if you got them on 1,000 BID versus 250 BID, the effect on the response is going to be different. Belatacept is a co-stimulation blocker and really paralyzes the ability to generate this response. And the reason this happens is belatacept actually paralyzes the T cell B cell interaction in the lymph node, because that interaction is co-stimulation blockade dependent. So what do you do with this, right? So this is from the Strasbourg group. They said, okay, what if we gave them a third booster? And so essentially what we start seeing is a slightly improved level of response. So you can see between the second dose and after the third dose, the antibody levels start going up. So that's where the additional boosters started coming up. This is from the Toronto group, and essentially showed the same thing. When you have an mRNA after the vaccine, you've got the third dose here. This is placebo. The level of antibody starts going up. Before and after the third dose, antibody in individual patients starts going up. Neutralization ability starts going up. Why would the neutralization ability go up? Because around this time, the variants are developing. Your breadth of antibody response is going up because of somatic hypermutation. This is from the French group, where you're looking at the third dose, antibody levels are going up. So this is where the CDC came in, and they have revised their recommendations for transplant patients. So this is specifically for the transplant patients. If you're going to give them the RNA Moderna vaccine, you give them the first dose. 4 weeks later, you give them the second dose. About 4 weeks later, you give them the third dose. This is part of the primary vaccination sequence for our transplant patients. Otherwise, they're not going to develop a response, or more than half of them won't. Then 3 months later, you give them a booster, and then you give them a second booster, and the booster can be homologous or heterologous. If you're using the Pfizer vaccine in adults, because the sequence is slightly different in children, you use the same sequence. If you're using the Johnson vaccine, the J&J vaccine, then what you do is you give them 2 first doses. This is your primary sequence. Then you give them 2 boosters. And it is preferred that if you're using the Johnson vaccine, the boosters be an mRNA vaccine. And then the Novavax vaccine, that's the protein delivery, only 2 doses. So, despite all this, there's going to be a lot of patients who are not going to develop a response. So, what do you do with them? If they can't generate antibody, we must give them antibody, right? So, monoclonal antibody cocktails have been developed. So, this is a study in which tixagevimab and silgagevimab, which is called Evusheld. I don't want to use proprietary names, but in this particular case, I'll make an exception because it's very difficult to pronounce the other 2 all the time. So essentially what they did was they took COVID-19 patients who didn't have antibody and gave them the Evusheld. Actually, this is the graph you want to look at. This is the graph in which they changed the scale on the y-axis to make the difference prettier. But essentially what you see here is that the amount of symptomatic infection in the treatment arm is lower than the control arm. So this actually provides some benefit to our transplant patients. This was studied when the Omicron variants were coming out. So these are the patients who received Evusheld. It worked against the Delta variant. It works against the BA1 variant. It worked against the BA2 variant. By that time, the BA5 variant, which is the current Omicron variant, was not in play. Very, very elegant experiment here by the French group. They took about 1,100 kidney transplant recipients and started sorting them out. Now, these are all vaccinated people, have got 3 doses of the mRNA vaccine. They found people who had actually developed a significant response, and they are now considered protected, and so that's your first group. People who did not develop a vaccine response were given different types of antibody, and there's some time sequence to this. So they gave them Evusheld, and they gave them some of the other antibodies which are not in play because they've lost their ability to deal with the Omicron variant. So people who received Evusheld or some of the other antibodies were considered unprotected. So what happens to them over a period of time? Symptomatic or asymptomatic Omicron infection. These are the people, the green ones are considered protected. So these are the guys who actually received vaccines and developed a response. These are the guys who received Evusheld. And then this is the line which shows people who were actually unprotected, did not respond to the vaccine and did not receive Evusheld. Same sequence in patients who got into the hospitals, patients who got in the ICU, or patients who died. So the level of protection Evusheld is providing is pretty close to that of successful vaccination. And so that's where the recommendations came by for us to give every transplant patient passive immunity as well to boost their chances of fighting off the infection. And this is the Omicron variants, and this is where things happen. You remember the RBD, we're all experts on RBD now. So all we are seeing is different types of mutations coming in the RBD, which makes it difficult for the antibody to attach to it. And once that happens, it becomes ineffective. And so we are trying to fight against that. So what happens when we give these different types of monoclonal antibodies to patients who actually have some of the Omicron variants? You've got the different variants listed over here, including the current BA.5. You give them tixagevimab. This is one of the components of Evishield. Some of the variants are knocked out. Silgagevimab doesn't work. Again, works against some of them, but not against all of them. But when you combine them, they take out a majority of the variants, and some of the other antibodies don't seem to work against the Omicron variants. So if I were to summarize the entire almost hour which I've been talking about in transplant candidates, pre-transplant, please vaccinate them, because this is the time when you know that they are very likely to mount a response. The response rates to 2 doses, especially 3 doses, in the dialysis patients is almost like that in the general population. More than 90% are eventually going to respond. That is the best time to give them immunity so that they have memory cells in their lymph nodes and plasma cells making sustained antibody in their bone marrow. But there's a bunch of people who will get to transplant and who would not have gotten vaccinated. There are some transplant centers who actually deny them transplant. They say you can't get transplanted. Our center does transplant them, but after transplant, as soon as we can, and depending on the antibody and the immunosuppression they get, in about 1 to 3 months, we vaccinate them, full series based on the CDC guidelines, and also give them passive immunity by giving them the antibody cocktail of tixagevimab and silgagevimab, Evishield. So that is the message. I think this is the message. Vaccinate them pre-transplant, but don't give up on them. If you get them post-transplant, they have not been vaccinated, please vaccinate them at the appropriate time, and then also give them passive immunity. You can give them the passive immunity cocktail about 2 weeks after the vaccination jab. And so this is the timeline of our response. I think it is a miracle that we had a vaccine being mass delivered in the United States a year after we found out what the virus was and what its structure was. This is, I think, one of the greatest endeavors and greatest achievements in the human history of fighting disease. We should all be proud of it. The battle and the quest continues. I'll stop here, and if there's any questions, I'll be happy to take them.
Microphones if anybody has questions.
Raise your hand if you have a question. I'll bring you the mic.
Can I ask, I don't think I need a microphone. Is there any negative effects of getting the vaccine after a transplant?
Yes. So that's a very, very good question. Why the stress on getting them vaccinated prior to transplantation is because of the figures I showed on the response rates, right? So essentially, the first thing is that when you vaccinate them before transplant, your odds of getting a meaningful response are almost double. That's one. Now, your question is pertinent that you're actually provoking the immune system. Could that lead to a rejection episode after transplant? We haven't seen that. So we have vaccinated pretty much with impunity, and we haven't really seen people developing an alloimmune component to that response. We have seen a lot of other autoimmune injury, but we have not seen an alloimmune response to vaccination. That's a very good question.
Good question.